Okay, this is chapter 11. Word of warning, this is, I think, somewhat more difficult than the last two chapters. Okay, so let's take this. So the first part of this is kind of give you a historical view. What came before our classical view of genetics? Okay, well, you look at some of the theories, basically we didn't know what was going on. Had a lot of theories. For example, some people thought that there was something called a homunculi, right? This is basically a little person and a sperm or an egg, and they would just come out like a pop goes the weasel. Well, did not happen this way. Okay, just to give you an idea that we just didn't know what was going on at the time. We had all these theories. And one idea was this kind of blending inheritance, which made kind of sense. Basically, it said that you take a tall and a short, and you're going to get an intermediate between the two. You cross a red plant, white plant, you get a pink plant. That type of thing. They saw that in some cases, right? But not all cases. So this kind of explains some genetics, but not all genetics. Okay, so we were basically grasping at straws about this. Then along comes Mendel. Now, at the time, his work was not recognized. It was rediscovered several years later. Okay, he died not knowing the importance of his work. Okay, so there you have it. Anyhow, his work gave us an idea of some classical genetics. Okay, so Mendel, you need to know the name. Other thing is what he did was he looked at plants and he collected indirect evidence about how inheritance works. He couldn't use a microscope, or he couldn't use all these fancy tools we have now to track the stuff like PCR and antibodies, all these things. No, he didn't have that. All he did was he saw the indirect evidence of how inheritance works. The indirect evidence. He watched for the effect. He studied this pea plant, and the thing is, this pea plant has a lot of good characteristics that make it well suited for this work. Okay. For example, this pea plant has some very simple genetics. It's not polyploidy. Okay, it's don't worry about that. Just know that it has a very simplistic genetic structure compared to a lot of other plants. Also, they's bred true, meaning that the traits he looked at, they were homozygous recessive or homozygous dominant. We'll talk about that later. But what you need to know is that he picked a very good plant for his work. That's good. Okay. Just talks about how he did his plants. Nothing too important here. Okay. What I'm going to do is I'm just going to go through these, and you have to know the terms. You have to know these terms, okay? Can't say any more simple than that. You have to know these terms, guys. Genes, locus, and allele. Let's talk about those. Okay. Genes are what? Heritable units of information. A locus is a particular location on a gene, and an allele is a particular form of a gene. How do I use those in a sentence together? Well, on chromosome 9, let's say that pink one is chromosome number 9, which is what blood type is on. Okay. Let's say P is an allele, and that lower P is an allele. See that P on the pink and P on the blue? Okay, where it's located is the locus. The allele is the big P and the big P. Those are alleles. Where it is on that gene, or where it is on that chromosome, is the locus. Okay, these are just things you got to know. Okay. Mutations, a change in the gene or the genetic structure. We'll talk more about that next section. Just know the bold terms, what those are. Really important terms here. Homozygous, heterozygous. Homozygous is when we're talking about one locus. Okay, one loci. One place on the two chromosomes we're comparing. When those are different, you are heterozygous. When those two are the same, you are homozygous. So let's look at this in this example. On the left, you see it says big P, okay? Where the P is on the pink one and on the blue one, that's the loci. Because we have the same allele represented by the same letter, we can say that that is homozygous. Little a, that's the middle one, okay? Same gene loci, same location on two homologous chromosomes. Because that's the same, little a, that's homozygous. In this case for recessive alleles. And on the far end, we have a big B and a little b. Those are different alleles at the same location. Those are heterozygous. Okay? When you see this picture, know how to identify a homozygous and a heterozygous. Talking about this later, but I want you to know that convention at the bottom. That's important. A big letter, capital letter, represents a dominant allele in a pair. Lowercase represents a recessive allele in a pair. That's important to know. Gene expression, that's just how protein is made. Okay, the next one, phenotype and genotype, very important. Genotype is the genetic makeup of an individual for two alleles for a gene. For example, if you have type AB blood, your genotype is A and B. You have an A allele and a B allele. That A allele and B allele is your genotype. So genetically, what alleles you have is your genotype. Now, how those are expressed is your phenotype. Your phenotype is something we can measure. Your height, your eye color, your blood type. Okay? If you're bald or not bald. That type of stuff is a phenotype. The alleles that lead to that phenotype is your genotype. Okay, think of gene, genotype. We're going to talk about a test cross later. Monohybrid cross is taking two alleles at the same locus and comparing them. Okay, so we're gonna cross two organisms, but we're just going to compare two one allele, or one gene. Know this. Each diploid individual possesses two alleles at a locus, and these two alleles separate when gametes are formed, one allele going into each gamete. Learn this, know this, love this. You will likely see this on the test. So let's talk about what that means, because that's a lot of words. Blah, blah, blah. Let's talk about that in English. Okay, English, what does that say? It says for every trait, we are going to have two alleles. One comes from our mother and one comes from our father. Okay, you got blood type, right? Yeah? Well, one allele for your blood type came from your mother. One allele for your blood type came from your father. Now, when we make our gametes, sperm for males, eggs for females, one of these alleles from either our mother or our father are going to find their way in that allele, in that gamete. You will not see a situation where both of those alleles find their way into the sperm or the egg, unless it's non-disjunction. We'll talk about that later. There is an exception. Okay, but that's an error. Okay, that's an error. So let's just say you have AB blood, and you have an A allele and a B allele. One of those comes from your mother, and one comes from your father. Okay, and when you make a gamete, it's going to have either an A allele or a B allele in it. Not both. Not both. Now, there is two exceptions to this. We kind of talked about it a little bit a second ago. One is in males, we have an XY non-pairing member, right? The Y chromosome is shorter, and there's no pair or counter to it. The other exception is non-disjunction. When DNA does not separate correctly, that's non-disjunction. Okay. DNA does not separate correctly during meiosis, it's non-disjunction. So at the end of meiosis, each of the gametes, the sperm or the egg, will have one copy of the allele. One gamete will not have two copies of the allele for the same trait. Okay, I'm not going to read all this because I'm going to talk about Punnett squares. Okay, basically this is all explained by Punnett squares. So Punnett squares calculate probability, and a probability is just a measure of a chance something's going to happen. So a Punnett square is how we determine the probabilities of genotypes and phenotypes. Okay, this is how we determine the probabilities or the likelihood of those appearing. So let's just say, in this example, you see the big A and the little a? Okay, let's say big A is tall, little a is short. Okay, big A is tall, little a is short for a plant. Okay. We're going to cross a male heterozygote because he has an A allele for tall and an A allele for short. So he has a tall allele and a short allele. And we're going to cross that with a female that's a heterozygote. She has a tall allele and a short allele. Now, if these, say these were plants, right? 1/4 of the plants they make will likely have a big A and a big A, meaning they are homozygous dominant. 1/2 of them are going to have a big A and a little a, meaning they're heterozygous. 1/4 of them are going to have a little a and a little a, meaning they are homozygous recessive. Now let's just say that they had 100 progeny, 100 plants that they produced. This says that about 25% or 25 plants will be big A, big A. About 50% of the plants are, 50 of the plants here would be big A, little a, and the remainder would be little a, little a. Now this is a monohybrid cross that Mendel did. Okay, we're crossing a homozygous dominant and a homozygous dominant, homozygous recessive. What happens? All the progeny are heterozygous. All progeny are heterozygous, meaning they have a big A and a little a. They have 2 alleles, a dominant and recessive. Now what we do is we take 2 of those. You see how it's big A, little a, and big A, little a? We take 2 of those and cross them. Okay, we're crossing 2 heterozygotes. When we cross them, what do we get? Well, 1/4 are going to have both of those big alleles, right? They're both of the dominant alleles, big A, big A. 1/4 will have little a, and 1/2 will have big A, little a. What this shows is a 1-to-3 phenotype. Okay, we have, well, the genotype is 1-to-1. For every one AA, there's 2, for every one big A, there's 2 big A, little a, and 1 little a, little a. The phenotype, though, here is if the big A is purple and the little a is white, then we have what? 3 purples for every 1 white flower. Now, Mendel used this in his monohybrid experiments. Mendel saw this 3-to-1 ratio over and over and over. And the only way he could explain it was through mitosis and meiosis. Now, you know, that is simplistic genetics we just saw, right? There's a lot of different types of genetic relationships out there. Okay. Many times it's not this dominant recessive thing, right? It's a lot more complex. So what's going to come next is some examples of this complexity. 4 examples we're going to look at are codominance, incomplete dominance, epistasis, and pleiopathy. So codominance is when you have 2 alleles that are not the same that are fully expressed. Each of them are expressed. There's no dominant recessive. They're fully expressed. Think of a white plant and a red plant. And codominance would be a plant with white splotches and red splotches, right? It's not a mixing. One is not in control of the other. It's red splotches and white splotches. So they're both being expressed. Another example is AB blood type. So if you have type A blood, you have A markers on your blood cell. Type B blood, you have B markers on your blood cell. Type AB blood has both. Incomplete dominance, it's a mix of the two types. One allele is not fully dominant over the other allele. Epistasis is when two or more gene products influence a trait. Okay, it's just not a one-to-one relationship. It's not like one allele makes tall and one allele makes short. This is something multi-gene. Finally, pleiopathy is when one gene has multiple phenotypic traits. So, for example, Marfan syndrome, that's one allele, but it makes multiple traits that are measurable. For this, you're going to need to know blood type. Blood type is just a monohybrid cross. Also, you have to know blood type. See that blood type diagram? You have to know those combinations of blood type. And also, if I was to give you a cross, you should know how to do blood type. We went over this in class. So, Mendel's law of independent assortment, or her second law. Genes on separate chromosomes are sorted into gametes independently of other genes. Okay. Now, this does not apply to genes on the same chromosome. So this is independent assortment. These genes are going to go independent. They do not care which ones go with which. They independently move around. It's the best way to say it. Also know this picture is, okay, here we go. Dihybrid cross. Dihybrid cross are when two traits are tracked. Okay, we looked at a monohybrid cross, right? We saw blood type as a monohybrid cross. Well, this is two traits. Now, the thing with these is, to get the ratios we expect, we have to have these genes on different chromosomes. If they're on the same chromosome, they're not going to behave how we expect them to. So, what we're going to do is look at a dihybrid experiment. Okay, dihybrid experiment, we have a big A, big B, crossed with a little a, little b. So this is one individual with two homozygous dominant genes, or alleles. The other individual is homozygous recessive. They have two recessive AAs and two recessive BBs. We do this cross, okay? Now, the gametes that big A, big B will form all are big A, big B. The gametes that little a, little b form are all little a, little b. We do this cross, right? And then what do we get? This is what we get. We get all carriers. We get all heterozygotes. Every individual will have big A, big B, little b. So then we do a cross with these guys. We cross big A, big B, little b with a big A, big B, little b. And this is what we get. A 9 to 3 to 3 to 1 ratio. Know that ratio. 9 to 3 to 3 to 1. 9 showing the dominant phenotype, 3 showing the dominant recessive phenotype, 3 showing the recessive dominant phenotype, and 1 showing the recessive phenotype. Linkage group, the further two genes are separated, the more often crossing over happens. They are linked genes. Continuous variation, many times when we have a trait that has many traits show variation. Okay, blood type is not one. You're either blood type A, you're type B, you're type O, you're AB. You don't have kind of A, kind of B, right? But some traits like eye color or height have a variation. There is a continuous variation in height. Okay, this is when we put this on a curve, when we line them all up into a statistical analysis, it all comes out as a bell curve. And that concludes this chapter.